Welcome back to Aging into Anatomy. With this presentation, we will discuss the physiological, functional, and immunological changes in the lungs with aging. A spectacular and recent review article discusses the cellular and molecular aspects of lung aging and failures in stress response pathway that contribute to development of pulmonary disease. Cells that comprise the airway and alveolar respiratory epithelium exhibit quantitative and qualitative deficits with age. Aging is associated with reduced mucociliary clearance in both upper and lower airways. Age-dependent telomere shortening has been observed in normal human lungs, but which cell types are responsible for these changes is unclear. There's this concept of senile emphysema where the alveoli enlarge. They're not destroyed, they just become more bulbous, and as a result, there's less surface tension and less recoil. Interestingly, as some studies show, there is an increased migration of macrophages. Part of this may be due to the accumulation of what we call the senescent cell phenotype. These are large fibroblasts that reside in tissue. They no longer have replicative potential, but they tend to exude inflammatory signals, thereby potentially increasing and recruiting more macrophages into the system, and that may be one of the theories behind the so-called cytokine storm that occurs with aging. The respiratory epithelium forms a vital barrier between the environment and internal human tissues, representing the first line of defense against inhaled pathogens, particulates, and other foreign material. One hallmark of aging is the depletion of adult stem cell reservoirs. Age-related quantitative and qualitative changes have been described for different progenitor populations in the lung. Basal and club cells decrease in number with age, whereas AT2 cells are unchanged in quantity, but exhibit deficits in self-renewal and differentiation capacity. Here's a table of different pulmonary changes. Notice the FEV1, forced expiratory volume in 1 second, declines with the age. Also note the VQ inequalities. There's lower ventilation in the lower half of the lungs, and for that reason, people as they get older need to be sitting up rather than lying down for optimal gas exchange. Here's an example of the FEV1 declining with age. Interestingly, there are some old people that look young, and some young people that look old, and so, there's again variation in lung function as you look at a population from age group to age group here from 25 years old to 75. With these pulmonary changes with age, the older adult, as you can see here, increasing age up to 80, the average pulse oxygen declines, which means they become closer and closer to a hypoxemic state. Cellular function is a composite of intrinsic biological processes and extrinsic interactions with other cell types and bioactive molecules. Both qualitative and quantitative changes have been described in specific cellular compartments in the aged lung. Red arrows represent decrease in cell number or in pathway activity. Green arrows represent increase in cell number or pathway activity. Thank you for listening to this presentation.